Evolutionary diversification is a much talked about issue in biology, particularly the causes and mechanisms of this adaptive process. A well-established and highly regarded theory suggests that adaptive diversification can happen as a result of ecological reactions between individual organisms, for example, competition and predation. However, recent observational studies have shown that diversification can occur within a sympatric environment with no geographical isolation. No examples have been found on a genetic level regarding this theory, until February 2013, when scientists Matthew Herron and Michael DeBelli from the University of British Columbia published an article documenting adaptive diversification occurring in sympatry on a genetic level. This was resulting from the competition for resources in populations of the bacteria Escherichia coli. Hence the title of their article, Parallel Evolutionary Dynamics of Adaptive Diversification in Escherichia coli. The question stands, can ecological settings lead to parallel evolutionary dynamics of diversification in different populations of the same species? Previous evolution experiments have shown that when exposed to a glucose and acetate medium, bacteria E. coli diversifies into two phenotypes that differ in the dioxic switch. That is the time it takes to switch from glucose metabolism to acetate metabolism. The slower switch takes a longer amount of time when compared to the faster switch. Herron and DeBelli used this to produce three different populations of E. coli, named populations 18, 19, and 20, each with a combination of both SS and FS clones obtained from day 156 of an E. coli evolutionary experiment. After these populations had grown in the medium, two large and two small colonies with SS and FS growth profiles were chosen for gene sequencing. As seen in Figure 1, the sequences revealed both parallel and unique genetic changes throughout the three populations, including changes to both genes and nucleotides. Independently evolved SS phenotypes shared mutations in the same gene, and in 4 cases shared an identical mutation in the same nucleotide position. Frequent time mapping of allele frequencies of the three populations also suggested that mutation is causing a specific physiology, change the environment, allowing the invasion of mutations causing an alternate physiology. Clearly, the FS phenotypes are better able to adapt due to their speed, allowing for a high diversity when compared to the SS phenotypes, as mutation frequencies are much higher. However, the parallel genetic changes seen in all sequences have provided empirical evidence of adaptive diversification as a novel evolutionary process.